Right, good morning. So today we will finish up with the DNA replication process, and we'll also start on the mitosis and look at what happens if errors occur and you're not able to control the cell division in the way that you are supposed to. Okay, so please take out this note that we started last class, and we'll add more details to it. Okay. So while you're doing that, let me just recap some of the key concepts that we covered. This picture is showing you the process of DNA replication, which is something that happens every time your cell divides. Okay. Before the cell splits into two, it must copy all of its genetic materials, and we are going to look at that process first in the prokaryotes, and then we're going to see how that happens in humans. So you started with the original parental DNA, and then you're going to create a bubble. Then you're going to expand the bubble horizontally and copy the DNA as you open up the bubble. So to create the replication bubble, you need to have the purple enzyme right here. And that is called helicase. So unzips the DNA double helix. And, I'm just going to add a little bit here and expands the replication bubble. So ultimately, the bubble will fully extend and separate the two strands completely. As this unzipping is going on, there will be some tension that builds up in the double helix. And you need to reduce the tension so that the strand's not going to break. That would be the function of gyrase. And lastly, because the DNA strands are attracted to each other, so you want to keep them apart so that you can continue to have access to the nucleotides. And that's what these orange balls are. They are single strand binding proteins. And sometimes they have a short form SSBP. And their job is to keep the DNA strands separated, prevents them from coming back together. Because if you close the DNA strands and you no longer have access to the nucleotides, you won't be able to build the new strand. Okay, so that's where the story went up to last time, and we're gonna continue with that right now. So I hope you brought more colors. Um, the next enzyme that is going to be important, I'm just going to put it on the side here, is something called DNA polymerase. Three. Okay, so we use Roman numeral 3. DNA polymerase 3. This enzyme is the main enzyme that's responsible for creating the new strand. The main enzyme for creating, we'll say for building, okay, for building the new DNA strand. It uses the parental DNA as a template. To build the complementary strand. Okay. So the black stuff here, that's the original, right? And we know DNA replication is semi-conservative. That means you're going to keep the original black nucleotides, and then you're going to use the base pairing rule to build the missing half. Okay. So, just give me a second, guys. Yesterday, my other class was like really confused about the whole semi-conservative, conservative, dispersive situation. So I drew this out for them, and maybe it's going to be helpful for you as well. You don't have to write this down. You can just look at it, or you can take a picture with your phone. So conservative means you take the original, and then you just directly make a copy of it, right? Semi-conservative, which is the correct one, means you separate the strands, the pink and the pink, and then you're going to build the missing half, the missing complementary half, right? So that is the enzyme that we're talking about, right? The enzyme that builds the missing half is DNA polymerase 3. Okay. So basically, when we add the suffix ase, it makes it an enzyme, right? Like helicase ends with ase, gyrase ends with ase. This is DNA polymerase. It's an enzyme that builds DNA polymer. And dispersive looks like this, right? You just take bits and pieces of the original randomly from each side, and then you fill in. So of course, as I keep on telling you, dispersive and conservative model is not correct. They're not correct. Right? Only semi-conservative. Who did what experiment proved that it was semi-conservative? Who were the people? You remember? Oh, Hershey and Chase is the other people. Okay? They show that it's DNA that's hereditary. The semi-conservative are these people. Okay, Messelson and Stahl. Okay? You don't have to memorize like the names. I'm not going to ask you who did what. Okay, but I'm going to ask you about their experiment though, on the quiz. Okay, back to yeah.
But the polymers that build DNA, what?
Yeah, so the enzyme that's building the missing half, yeah, DNA polymerase. So it's an enzyme that builds DNA polymer. Okay? So DNA polymerase, and this is number 3. Okay, it turns out there are other ones as well. Yeah, so that's that. Now this guy, this DNA polymerase, has 2 problems, 2 limitations, we'll say. Limitations. Okay, number 1, cannot, it can only build on existing DNA. Can only build if there is already something in place. Okay? Think of it like this. Don't copy this down, but think of it like this. Okay? It's like, I think of my kids, okay? Like they come home with homeworks, whatever, like my pen's not working. The younger one is doing multiplication. Oh, pen's not working, gotta wait. So it's like that, right? So come home with like whatever multiplication sheets. Okay, and then she just sits there and not do anything. Okay, and then I have to sit down with her. I'm like, okay, 2 times 4 is 8, 7 times 8 is 56, and then she's like, okay. And then because a few questions done, she can finish the sheet herself. Right? That's DNA polymerase 3. DNA polymerase 3 is my kid. They're not gonna build anything. They're not gonna do the sheet until someone does a few for them. Understand? That's problem number 1. Okay, I'll tell you how to solve it in a second. Problem number 2. Fingers crossed. Hopefully my pen works now. Oh, it does. Okay, so number 2. Oh, but it won't let me change color. What is going on? And give me a second. Let me restart this. All right. Problem number 2. Can only build in 5' to 3' direction. Okay. And to be clear, I'm talking about on the new strand. Okay, remember, DNA has directionality to it, right? One strand is 5 to 3, the other is 3 to 5. And so when we are creating the new strand, it needs to be in the direction of 5 to 3. Okay, it doesn't know how to go the other way. So those are the two big, those are the two big limitations associated with DNA polymerase 3. Okay. Any questions? All right. So we have the replication bubble here. We have the original parent DNA, and we want to make the missing half. We want to build a missing half. But like I said, the DNA polymerase 3 does not want to start on its own. It cannot start without someone doing a few nucleotides first. Okay, and that's where another enzyme is going to come in. Let's use my favorite color. Okay, so there's going to be an enzyme called and you can use whatever shape you want. Okay. There's going to be an enzyme here, and this enzyme is called primase. Primase. Okay. So primase is like me. Right? I'm gonna sit down and do a few questions for my kids first, so that they could continue on their own. So primase is gonna come, and it's gonna look at the parent DNA, and there's a C here. So what nucleotide should we put to build the new strand? Yeah, it's not a trick question, guys. G, right? Okay, G. There you go. Okay, and then they see T. What are they gonna put? And then A, right? And then this is the tricky part, right? When they see A, what are you supposed to put? Normally, what are you supposed to put? T, right? Okay. But the primase is building RNA instead of DNA. So instead of instead of pairing A with T, is going to be pairing it with U. Okay. So this thing that we just built, this is called an RNA primer. Okay. It's just so that DNA polymerase 3 can continue to build. Okay. Now that there are some existing nucleotides, it can continue to build on that. Okay, so this is RNA primer, and it's laid down by the primase. So primase will create a few RNA nucleotides so that DNA polymerase 3 can continue. Do you understand why we need this extra step? Right? Because the enzyme will not do it on its own, right? That's the biggest problem. Understand? All right, this next part is really important. So I need you to know the answer for certain before we move on. Look at the black strand. The black strand is the original DNA. And let's take a closer look at its direction. 5' to 3', right? So when we build this new strand, the primer here, what number should I put over here? Should I put 5' here or should I put 3' here? 5 or 3? What do you guys think? Show me, 5 or 3? Okay. Anyone else? I saw 3 answers. Anyone else? You got to know whether this is 5 or 3 for the rest of the note to make sense. Okay. It's 5', right, guys? Okay. Because it's anti-parallel. If you see the black being 3 here, this has to be 5. Okay? And then that means this little bit over here, that will be 3'. Okay? That takes us to the second limitation of DNA polymerase 3. Second limitation, I'm just going to read it to you again, can only build in the 5' to 3' direction. Sorry, missing a few letters here. Direction, ION, on the new strand. So when the DNA polymerase 3 attaches, is it going to continue building over here, or is it going to continue building over there? Okay. Which way is the correct way? Towards the fork or away from the fork? Towards the fork or away from the fork? Based on what we wrote down here. Can only build in 5' to 3' direction on the new strand. Towards the fork. Put 1 for towards the fork. Put 2 for away from the fork. What do you think? Okay. Undecided people. Okay. Okay. 5 to 3. This is 5. This is 3. This is the new strand towards the fork, right? Understand? It can only build in the 5 to 3 on the new strand. So we're not looking at the numbers for the black. We're looking for the numbers on the new one. Okay. So when polymerase 3 comes, it's going to build it like this. It's just going to continue. Okay. C, G, G, C, G, so on and so forth. Okay. So we're going to have an enzyme that does that. It's getting crowded. Okay. But this would be DNA polymerase 3. Do you understand? Now, I hope you start to see a problem. Okay? Remember, the bubble is like this, right? We're only focusing on the one side of the fork. So that means we still have a bunch of nucleotides here, right? So if the polymerase can only go that way, how are we going to do the rest of them, right? It's like, you know, the math worksheet analogy. Okay? Let's say the worksheet is really big. Okay? And then we start in the middle of the worksheet, okay? And then you only go down. Okay? So how are we going to do the rest of the sheet, right? Okay. So the answer is like this. I will have to do a few other questions here. All right, 9 and then 28. And then you can go down. Do you understand? So the problem is that you cannot go back. The problem is you cannot go in the 3 to 5 direction. You can only go 5 to 3. Okay? So that's why, like, to copy the rest, we're gonna need to have more primers. And that's actually is what's going on the other side of the fork. So listen, I'm gonna need another primer here. And it's gonna be like GAUU, right? Okay. In real life, it's much longer, but that's fine. We'll just do it like this. And can you please tell me what number goes here, 5 or 3? Over here, 5 or 3? 5, very good. Okay, because look, this is 3, right? 3, 5. It's always different, huh? And that means this is 3. So that's a RNA primer. Keep looking. When DNA polymerase 3 comes, is it going to go that way, away from the fork, or towards the fork?
Wait,
Away from the fork, towards? Away from the fork, right? Because it can only build from 5 to 3, right? It can only do the worksheets down. Understand? So polymerase 3 is going to come. G is going to go with C, G, A, and so on and so forth, right? But then we can see the problem now. What about the rest of this stuff? As we unzip more, as we open up the bubble, how are we going to copy all this stuff? Like, that's fine. That's just going to keep on going. But what about this? Well, like I try to tell you over there, you're going to have to put another primer now. Okay? And then polymerase 3 will be able to do it like that. And then as you open up more, you're going to need another primer. And then polymerase 3 will do that. You sure you're okay? Yeah. Like you might ask, why don't we just wait? Why don't we just wait until we open up enough and then we put one primer and then just go all the way, right? Okay, like that's a sensible thing to do, right? But you have to understand the DNA here is very big, right? We talk about million of base pairs. If you wait until the whole thing unzips and then start building, the process is gonna take too long, right? So you gotta do it as it's happening. As you unzip, you gotta start building, right? Bless you. And until the end, you're gonna be able to do it. You're gonna be able to finish the job much quicker than waiting it to be the end, right? Okay. All right. I am just gonna wait here for you to copy what you need. And then we're gonna start a brand new diagram to summarize this thing. And then we're gonna move on. Now, you might wonder, like, why does anybody care about this thing, right? Actually, do you guys remember PCR testing during COVID? You do a nasal swab, collect the sample. You can do an antigen test, which is testing for viral proteins, or you can do a PCR test, which tests for viral DNA. The technology of PCR is actually based on the understanding of this process. Okay, and we actually will learn about PCR in lecture 9. Okay. All right, so I hope you have a nice picture of this thing. If you don't, that's fine. I'll post it online and you can do it nicely when you get home. All right, so let's summarize. Okay, overview, I guess. Overview of the entire replication bubble. Okay, so draw your best bubble. We're not going to put letters in it. 5 to 3 on the top. And again, I'm just randomly choosing 5 to 3, right? There's no reason why 5 to 3 has to be on the top. It could be the other way around, but I'm just being consistent with all the other diagrams I've drawn. What about here? What number do I put here again? Now 3 to 5. Okay, so we were looking at the left side of the fork, but basically the idea is this. You're going to put a primer here I'm just gonna label it for you one more time. This is an RNA primer. And polymerase 3 will go this way. Okay. And remember, 5 to 3. Nice. So this DNA strand is basically going to be produced continuously. And as you unzip more, it's just going to keep on going and going and going, right? So this is called the leading strand. Leading strand. It's built continuously towards or into the fork. Leading strand built continuously into the replication fork. At the bottom, you're going to have a primer here. And, just think about the direction. So the black one is 3 on the left, which means the new one is 5 here, and then 3 here, right? So the DNA polymerase is going to go this way. So this is going to be the leading strand on the right side. Now, as you open up more of the fork, you're going to need more primers. And then you basically will have to build more and more. So to contrast the top half with the bottom half, the top is the leading strand because it's being built continuously, but the bottom is being built in fragments, right? Yeah. So that is called the lagging strand because it's a bit slower to do it in fragments. So it's built discontinuously. Okay. In fragments. Away from the fork. Now these fragments have a name. Right? It's named after the scientist who discovered them, I guess, Okazaki. And then on the other side, you would have more fragments, right? So of course, on the other side of the bubble, this would be the lagging strand. Any questions?
Seems like an
What's that? Yeah, it's a mirror image. But
What?
yeah, let's flip it. I'm not gonna ask you to draw any of these on a test, so don't worry. But you obviously have to know the enzymes and then all the stuff. Anything you want me to clarify before we continue? Or like three more things I have to tell you before the note is over. Anybody? Okay. Now we're gonna zoom in, zoom out all the time. And we're gonna go over here. We're gonna take a look at this part, and then we're gonna zoom in. Okay. So I'm just gonna randomly make some letters up. And so the pink stuff here, that's like the primer. And then the rest is gonna be filled up with the DNA polymerase, right? Now this part is really hard for me to show. Um, I'll try. Okay, what first, and then you copy after. RNA primer, right? And then the DNA polymerase 3 will come and build the nucleotide, right? So it's gonna go this way. All right, so it's gonna go this way. Build. Okay, these are all nucleotides. I just don't have the letters, and then it's like A, T, G, T, C, C, G. Okay, just watch first. Okay, so this is DNA polymerase 3. So it's just gonna zip along, gonna put the nucleotides on. So far, so good, right? When it gets to the primer, that's ahead of it because the primer was laid down like before, right? So when it gets to the primer, then it falls off. Okay? So number 3 is gonna fall off, and then it's gonna call DNA polymerase 1 to come over. The number 1 will take over, and then it will cut away the RNA primer and replace it with DNA. Because remember, the RNA primer has U, right? Uracil. You cannot have uracil and DNA, right? You understand that? Okay, so like low-tech animation. Number 3, build. When it gets to the primer, it falls off, and then it calls number 1 over, and then it will finish the job. With me? Okay. Now let's draw that. So as DNA polymerase 3 approaches the RNA primer in front of it, I guess. It's replaced by DNA polymerase 1. Okay, so this guy falls off. And then number one's gonna take over. Okay. DNA polymerase 1 will replace the RNA primer with DNA nucleotides. Okay, both polymerase 1 and 3 has spell checking and autocorrect ability, meaning as it's building, if it accidentally put in the wrong nucleotide, it will just move back one space, take it out, and then change it. It's like typing. Right? You type, and then you realize you made a mistake, you press backspace, and then you correct the mistake. Right? So this way, it can minimize the chance of introducing errors and mutations into your DNA. Right, I just have one last thing to tell you, and then we'll be done the note. Well, I have to upload it afterwards, right? Yeah, they will be there. All right. Finally, there's one last enzyme. I'm not gonna put it on the picture. It's just gonna be like way too much already. So I'm just gonna draw it out for you, write it out for you, I guess. Finally, lastly, the enzyme. I'm running out of color myself. The enzyme, let's use brown. DNA ligase. Okay, to ligate is to join things together. So this is an enzyme that joins things. DNA ligase will connect the Okazaki fragments. Is that even the right color? No. I think I used pink before. You know what? I'm just gonna use black. Connects the Okazaki fragment. Huh? And that's it. That's how DNA replication occurs in prokaryotes. That took like decades of work to figure out. Now, remember, I told you prokaryotic DNA are circular. Okay. So it looks like this. Now, obviously, it's a double strand, but I'm not gonna draw that because it's like confusing to draw double strand. Just watch. So basically, you're gonna have a primer, and then you're gonna build this way. Okay, and then you're gonna like have another primer, another primer, another primer, and maybe another primer here. So it's gonna build all the way to the next primer. I just want you to be able to understand this, right? So it's a one full loop, right? Whenever you get to the new primer, three is gonna fall off, number one's gonna come, and you're gonna replace the pink bits. And eventually you go full circle. Okay? That's what happens in prokaryotes. Do we have circular DNA? No, we have linear DNA, which, as you will see, is going to be quite a bit of a problem. Okay. Last call. Any questions? I know I keep on asking you every 5 minutes, but I need to be sure. Don't be shy. If you have a question, I guarantee someone else has the same question. Well, you have my email if you want to ask me later. All right, so this is just like the nice picture, right, of what we were trying to do. You probably are able to identify some of these things. It's probably a good idea to check it out yourself. Okay. Although there is an error in this picture. If you follow it, it's 5 over here. And if I keep on going and going, okay, and then they did the twist here. I don't know why, but if you follow the twist, then that's actually wrong, right? I just realized. So see, this is supposed to be 3, and this is supposed to be 5. Okay, you know what I mean? Like if you follow the 5 all the way to the other side, then it makes sense, right? It's not a big deal, like, but just in case, nobody ever figured out that mistake unless I point it out. I should change it probably. What are these things anyways? Like maybe we just try to figure out a few of these things. Like what do you think number 2 is? Look at the notes. Look at the ones we drew. What's helicase, right? Okay, beautiful. This one? Gyrates, yeah, and these little balls. Exactly. Right. Okay. I'm not going to ask you to label it on the test, okay? But like, again, the diagram should help you conceptualize what's going on. All right, so now we're gonna talk about DNA replication in eukaryotes and the problems that we have. All right, it's gonna get quite interesting for real, I hope. So, prokaryote DNA replication differs in a few key ways compared to what happens in us. Number one, our DNA is linear, and it's much, much longer, right? For something like E. coli, which is prokaryotes, they have about 40 million base pairs in total. Okay, 40 million ACTGs, right? For us, all of our DNA, we have about 3 billion base pairs. So it's much longer. And for that reason, it makes more sense to have multiple origin of replications at a time. So instead of just having one bubble and then spread out, you're going to have multiple bubbles, and then each bubble will expand, and eventually the bubbles will merge, right? So you start at multiple sites. That way you can copy many different locations simultaneously and you speed up the process. Okay. So that's the first difference. We have multiple origin of replications. Prokaryotes only have one. Number two, most of the enzymes in prokaryotes have a eukaryote counterpart, but some are represented by multiple enzymes. So, for example, in E. coli, there are only DNA polymerase 1 and 3, but in humans, there are 5 different ones. Okay, there are DNA polymerase 2, and then a few other ones that exist. Okay, but for the most part, they do the same thing. And finally, the most important point that we're gonna actually talk about for the next 15 minutes, some DNA are lost in us every time we make a copy of it. Okay? Every time we duplicate our DNA, we lose a bit of the DNA at the end. Okay? And then why? So I'm going to try to explain to you why that is the case. All right, back to our note here, just briefly. We're going to have linear DNA in eukaryotes. Okay? E.g. humans. Human cells. So just going to do 5 to 3 again, and then 3 to 5. By now, you should hopefully be familiar with the process. To copy this thing, I'm going to need a bunch of primers, right? So that's what I'm going to do. I'm going to put a bunch of primers. Primers. Please let me know if you have a primer here. Take a look. If you have a primer here, which way will the replication go? To the left or to the right? Please think about it. Point to it, left or right, sorry. Okay. Not 100%, huh? Okay. Think about it. Again, look at the numbers. Okay. Which way is it gonna go? I'm talking about the top strand here. After you put the primer, which way will it go? Left or right?
Understand, right? So
What about audience at the back? What do you guys think? Okay, look. What number do I put here on the right side? Help me. What number do I put here? 5 or 3? Huh? 5, right, guys? 5, right? Because if this is 5, then this is 3 on the black, and it'll be 5 here, right? And then this is 3, right? So I asked you again, do I go left or do I go right? Because the DNA polymerase can only build in 5 to 3 on the new strand. Okay, so it's gonna go this way. You absolutely have to understand that before we go on. And if you don't understand, just look at me, slowly shake your head, I'll explain it. Imagine how difficult this is to teach online in the afternoon, right? Nobody turns on their camera, just talking to myself for 2 hours. I have no idea if they get it or not. I don't actually know if you guys get it, even though this is live audience. Okay, how about this one? Left or right?
left.
It's left again, right? Okay. So it's gonna go this way. Okay. And it's gonna go that way all the time for the top strand. If it's 5 to 3 like that. All right. Now there's a point to this, okay, and you will see in a second. I'm gonna just draw the same thing at the bottom. The primers might be at a different location, but it doesn't really matter. Now, I hope you understand this is going to go to the right, right? Okay, I'm going to repeat myself one more time, but watch and listen. What enzyme builds the blue strand? Polymerase 3, right? DNA polymerase 3. When it gets to the ping, when it gets to the primer, 3 falls off and number 1 comes on, right? This, look, guys, this is going to get replaced with DNA, correct? This is going to get replaced by DNA. That's going to get replaced by DNA. That's going to get replaced by DNA. However, this will not be replaced by DNA. Why? Because there's nothing coming from above. Okay? This is linear. It's not like the circle case of the bacteria. In the bacteria, you make a full loop and you're able to loop back to your starting point, right? However, with us, with linear DNA, there is no number 3 coming from upstream, we say. And if number 3 is not coming, there's no signal to tell number 1 to come on. Okay? So this little bit of DNA, this little bit of RNA is never going to be replaced by DNA, and it's going to be gone. Okay? So you lose a bit here, and you lose a bit here. This happens every time we copy our DNA. Okay? Every time your cell divides, you copy your DNA, you lose a bit at the end, and you lose a bit at the end. Imagine every time you read your textbook, you have to rip out some pages from the end. Okay? Eventually, what's going to happen? Your book gets thinner and thinner, right? And you're going to start losing important information. Okay? Nobody really cares about the glossary or the table of contents. If you rip those out, right? The book is still functional for the most part. But if you start ripping out the actual chapters, then you lose critical information, right? And that's what happens here. If we start chopping the DNA a little bit at a time, eventually we're going to damage the genetic material that codes for the proteins. Remember, DNA has the instruction to make the proteins, right? So if we start chopping those off, then we're going to have a problem. Okay? This is only a problem for humans, for eukaryotic cells, but not for bacteria, right? Because they have circular DNA. They can loop back to the starting point and replace their own primer, but not for us. Okay? So to delay this process, to allow our cells to divide as many times as possible without eating into the actual useful genetic material, we have this special protective sequence that are located at the ends of the chromosome. This protective sequence is called telomeres. Okay, telomeres. They are non-coding DNA that are located at the ends of the chromosome, and they are just repeats of TTA-GGG. Just keeps on repeating. TTA-GGG, TTA-GGG, repeated like a couple hundred times. Right? So what that does is, when you are replicating your DNA, instead of chopping the actual coding DNA, you're just chopping off the telomeres. Okay? The telomeres is like the plastic thing at the end of your shoelaces. Right? It protects the shoelace, so it's not going to fray open. Okay? But here is the metaphor, right? Analogy. Every time you tie your shoes, you have to cut off a little bit of the plastic thing. Okay, so you can cut it off. It's still going to be okay. Eventually, you're going to run out of the plastic thing, right? Okay, and if you cut it again, then the shoelace is going to start falling apart, right? So that's what happens. Our cells will divide, and every time it divides, every time it copies its DNA, you will lose a little bit of telomeres on both ends. And if you lose enough telomeres, if your cells have gone through enough number of cell divisions, the telomeres will become critically short. And then the cell will no longer be able to divide. Okay, it's not allowed to divide anymore because if you do go through another round of division, you will end up losing important DNA information. Okay, so most cells can go through like 30, 40 rounds of divisions, and then it will not be allowed to divide anymore. Okay, the cell becomes senescent. We say it's old now, and so as we age, the pool of senescent cells are going to increase because they've gone through many rounds of division, right? And that is what contributes to aging. You with me? Okay, I have a question for you, and then we're gonna take a break. Now, I don't know, most of you are, I'm guessing, early 20s, late teens, maybe. Okay, so you've gone through 20 years of cell division in your life, right? Okay, I'm in my 40s, so I've done like twice as much division as you. Okay, so when you're gonna have a kid later on, if you choose to, okay, let's say you have a kid at like 30, right? Okay, so when you have a kid at 30, by that time, your cells have gone through divisions for 30 years. The telomeres are quite short compared to when you first were born, right? Okay. So when you have a kid, how come your kid is able to grow and go through another 30 rounds or 40 rounds of division in their body if they inherit short telomeres from you? Do you understand my question? Let me do that again. Right. So this is your telomeres. This is your DNA when you were born. Okay. As you grow, shorter, right? Okay. Now you want to have a kid. Right? You're gonna pass this on to your child, right? So won't they start with short telomeres, and then when they grow up, it's gonna be shorter, and then next generation, shorter, right? Okay. So if this keeps on going, then no more kids, right? Humanity is gone, right? Okay. You see the problem? Okay. So obviously that's not true, right? Okay. But it's a valid question. Okay? And the reason why that is not true is because there is an enzyme called telomerase. Telomerase. Okay? So telomeres are the repeats that are at the ends. Telomerase is an enzyme that will re-lengthen the telomeres. It will make it long again. But it will only do so in certain kinds of cells in the body, specifically in germ cells. Okay? A lot of people get confused. They hear germ cells, they think of like bacteria and whatnot. That's not true. Okay? Germ cells in the context of our body, these are cells that will become sperm or egg. Right? In female, it's going to become eggs. In male, it's going to become sperm. Sperm are made in the testicle, and the eggs are made in the ovaries, right? So those are the places where you will have the germ cells. So in those germ cells, the telomerase will reset the telomeres before you pass it on to the next generation. Okay, so even though you have kids, by the time you are 30s, 40s even, right? Even though your telomeres are short, but they get reset before it's passed on to the next generation. Do you understand that? Okay. Now here is a question for you to think about during the break. Oh, sorry. Here's a picture of what it looks like without telomerase. It gets shorter and shorter, right? And then critically short. Can't divide anymore. Of course, it's gonna be much more division than what's shown here, but you get the point. With telomerase, it's gonna be long, right? Okay. So rewind 10 minutes earlier. I was telling you, when as we grow, a lot of our cells will have much shorter telomeres, and eventually it's going to get to a point where they're not allowed to divide anymore, right? Okay, and that contributes to what? Aging. You with me? Okay. So here's the logic, right? Some people are saying, sometimes they would think, then why not like give the cells telomerase? Right? If we give them telomerase and then they reset the length, and then won't that like rejuvenate the body? Right? If our cells are no longer senescent, if they're no longer prevented from dividing, maybe we can reverse the aging process, right? Do you guys follow the logic? Yeah. Okay. I'll let you think on that during the break. We'll continue afterwards. What time is it anyways? Let's see. Oh, perfect. 9:30. Okay, so we're gonna have a 5-minute break. Okay. And then we'll do more right after.
Minutes.
Okay, so let's keep going. Um, this picture here shows you the location of the telomeres. Right? Each of these X's, right? Those are chromosomes, and the part that lights up at the end, those are the telomeres. If there is a technology that you can use, it's called fish, that you can make a specific part of the chromosome lights up. Okay, so in this particular case, they want to light up the telomeres, and it looks like that. Okay, so before we continue, let's take a moment and talk about Dolly. Anybody heard of Dolly? Yeah. Okay. Dolly was a famous sheep because she's the first mammal to be cloned. Okay. So clone means genetically identical to the mother, right? Okay. So it only lived 6 years, which is half of the expected lifespan of a sheep of that kind. So let's talk about the story of cloning Dolly. Cloning Dolly. It's called nuclear transfer. Okay, just random fun question. How many sheep do you think it takes to clone a sheep? Not all the ones that failed and die, okay? Like the successful case to make Dolly, how many sheep do you think it takes? One, two, three, four, five. Show me. What do you think? Okay, 2 sheep. Anyone else? Just random guess. There's no right or wrong. Yeah. Okay. 4 sheep. Yeah. Okay. All right. So let's find out. Okay. You guys are no fun. It's so boring. So we have a sheep right here. Okay. So actually, I don't think it was too happy with the to be in the experiment. Okay. There we go. This is egg donor. Egg donor. All right. So they're gonna take the egg, an egg. Now, sometimes in the past, people ask me. One time, a student was really puzzled. Was like, "Sheep lay eggs?" I was like, "No, no, no, not like chicken egg, like like eggs." I was like, "Oh, okay." So this is like like we have eggs too, right? The females, you get it? So like they take an egg from the sheep, and remove the nucleus. Okay, so this is just like if you would, it's like the eggshell, right? Just the egg cell with no nucleus. Okay. Normally, an egg would contain half the genetic material from the mother, right? Where's the other half coming from? The dad, right? The sperm, right? Okay, so normally an egg would have half the genetic material. The sperm fertilizes the egg. Half plus half gives you back the whole thing, right? So here they remove the nucleus. So it's just a shell. There are no genetic material in it anymore. All right. Okay, that's sheep number one. And we have sheep number two. All right, so this lady, actually, this female sheep is going to be the nucleus donor. So take a somatic cell from the sheep
She's
and no one's pregnant yet, and extract The nucleus. Our body and the body of a sheep, okay, of animals, there are two kinds of cells: somatic cells and germ cells. Germ cells, as I told you, are cells that will become sperm or egg. Somatic cells are everything else. Okay, so like your liver cell, your kidney cells, your brain cells, skin cells, all those cells are somatic cells. So they took a somatic cell, I forgot from where, from the sheep, okay, but I don't know like what place, it doesn't matter. And they extract the nucleus from it. Okay, so that's gonna be just the nucleus. The nucleus contains both sets of chromosomes, right? Both set of genetic material, as opposed to half for the sperm and egg. Right? So that's the story so far. You guys follow? All right. What do you think is going to happen next? What are we going to do with the shell and the nucleus? That's right. They're going to put the nucleus into the eggshell. And for those of you who wonder how that happens, they literally do it under the microscope, okay? They like inject it into it. You can find YouTube videos of that. And you've probably seen it on like movies or something like that, okay? That's how people do like in vitro fertilization. Anyways, so then the nucleus is going to go into the eggshell. Combined. And there we have a fertilized egg now. Okay. But no male was involved in this process, right? Okay. It's fertilized because it has full set of chromosomes. But the whole set of chromosomes is donated by the nucleus donor, right? You understand? There's no male sheep involved here. Okay. So 2 sheep so far. Now we need a 3rd sheep. What do you think the first sheep is the job is? What do you think this one is gonna do? That's right, they're gonna put the egg in this sheep. So what do we call people who carry babies for other people?
Surrogate.
Surrogate, that's right. So this is a surrogate sheep. Okay, and then you have Dolly. All right. Now, unlike movies, if you actually gonna clone, let's say you clone me, right, for whatever reason, okay, clone me, it's not gonna be like a grown up 40-year-old me when you clone me, okay? If it does happen, it's gonna be like the baby version of me, right? And then it's gonna grow up, not even necessarily looking like me, okay? Because even twins, when they grow up, they don't necessarily look identical anymore, right? Okay, but it's gonna be genetically identical to me. So in this situation, Dolly, it's a clone, but it's gonna born as like a baby sheep. You with me? So here's my question. 3 sheeps involved in making Dolly. Is Dolly a clone of number 1, number 2, or number 3? Show me your fingers. It's a clone of which one? Okay. Yeah, most of you say it's a clone of number 2. That's correct. Because number 2 is the sheep that provided the genetic material, right? And the eggshell has no genetic material in it. Right? The surrogate is just a vessel, right, to carry the baby to term, right? Okay, so genetically identical to number 2. Okay, so why are we talking about this? Remember, Dolly only lived 6 years as opposed to 12 years, right? Okay, and that's not because of some technical failure in terms of the cloning process. Okay, the reason why I only live half as long as an actual sheep is because of the telomeres. Okay, the nuclear donor when we took the cell from the nuclear donor from sheep number two is an adult sheep, right? So at that point, its cell has been going through division for some time. So its telomeres are already shortened. So when they transfer the nucleus into the egg and then grow the shell, grow the sheep is born as age 0, like a baby, but genetically, its telomeres are already shortened quite a bit, right? And so it age very rapidly. Does that make sense? Okay. So why this is not a problem for normal sheep? Well, normally sheep reproduce with the sperm and the egg, right? And every time we produce the sperm and the egg, we reset the telomeres length because of the enzyme telomerase, right? So that wouldn't be a problem normally. But when you clone it using this method, the nuclear transfer method, the offspring, right, genetically is going to be old, even though biologically it's young. Okay. All right. So back to our question. If telomerase and telomeres seems to play a role in the aging process, and I asked you at the time before the break, wouldn't it make sense to, maybe try to use telomerase as a way to slow down aging or even reverse it, right, in some instances? It turns out that will not work because cancer, which by definition is uncontrolled cell division. Normally, our cells would divide up to a point, and then the telomeres keep it in check, and it stops it from dividing further if it has gone through cycles of division. But in cancer cells, one of the properties of cancer is that they can virtually divide indefinitely, okay? And one of the reasons why it can divide indefinitely is because they have telomerase, okay? They are not supposed to express telomerase. Only germ cells are supposed to have it, right? But cancer cells, because of mutations, they are able to have telomerase. And that allows them to have really long telomeres. Okay, every time it gets too short, they just make it longer. And it can divide and divide and divide. Okay? Yeah. Okay, so we're going to learn more about that in a bit, but short answer is cancer are basically from our own cells.
Answer fitting in, is this so random?
No, it's not random.
No.
Because I was telling you, right? Normal cell, normally the cell divides and then divides, right? And then it divides, right? Okay, so like you get a bunch of cells. Okay, so up to this point, it has gone through many divisions already, and then it needs to stop. Okay, so that's what happens normally. In cancer, they do not stop because of the telomerase. Okay, there's the telomeres don't get critically short, and that stop signal is no longer there. So even after it's gone through like 40 rounds of division, it's going to keep on dividing. And when the cell keeps on dividing and dividing and dividing, you end up having a tumor, right? Does that make sense now? Okay. So even though telomerase is critical in reproduction because we reset the length, okay? But having it in the wrong cell is actually quite bad because it could create cancer. Okay. So one of the treatments that they are exploring in treating cancer is telomerase inhibition. Right, so if they could target the telomerase in the cancer cell, that would put the brakes back on, right? It would stop the cell from dividing and hopefully, shrink the tumor. But of course, there are severe side effects to that, right? If we inhibit telomerase, if we don't do it in a way that's really specific, it might end up causing the person to not be able to have babies, right? Okay. You guys able to connect all these things together, hopefully? Yeah. That was a great question, okay, by the way. Okay, concept check questions. Yeah, that's good. Yeah. Anyone else? Yes, good. Good. Last call. Not quite. Number two, right? B. Yeah. Prokaryotes only a single replication bubble. We have many. Because I told you we have billions to copy, right? So it makes sense to open multiple bubbles and do it simultaneously. Okay, A is a lot of people chose A, but that's not true. Okay, prokaryotes do not need telomeres because their DNA are circular. They loop back on themselves. Right? We only need telomeres for linear DNA. How about this one? This one should be quick. Good, good, good. Uh-huh. Anything? Okay, that would be ligase, right? Very good. Ligase. Finally, last one, I think. Good. So it's, a lot of the questions will be like this on the quiz. It's like just straightforward. There will be some where you have to think like that, okay? But the answer is DNA polymerase 1. Oh, another question. One more. Um, Not quite. Okay. Yeah. Okay, good. Anyone else? Yeah. Very secret. Yeah. Correct. Okay, telomerase is needed to re-lengthen short-term chromosome, right? It resets it. Okay, it does not prevent cancer. It actually contributes to cancer. Okay. All right. That is that. So you guys need a moment, maybe just a couple minutes, just breathe, okay, and then we'll continue. Okay? Just like sit there and chill a little bit, couple minutes.
Let's go.
It's okay.
So cancer cells are basically a
Yeah, essentially like error, right, in the DNA. Okay, so that was the first half of the lesson, first half of lesson 6. Now we're gonna do the second half. Second half. Next week we probably won't be able to finish 6 today. We'll finish number 6 next week, but we'll also start on 7. 7 is a lot of work, okay, like calculation work, and it's like math and chemistry where you have to do stuff, okay? So please print the PowerPoint slides as well as the study guide. Okay, you're gonna need both of them to do in class. Okay. All right. So second half is cell cycle and mitosis. Let's revisit the cell theory again. One of the 4 parts of the cell theory states that all living cells come from division of pre-existing cells. And we know that already, right? It needs to divide. And so before it divides, we need to copy all of its genetic material. And the reason why cells need to divide is that, number one, you need to replace lost or damaged cells. If you have a cut in the finger, you lost some cells, you need to replenish them, okay? We also need cell division for growth and development. You have many more cells now than when you were first born. And for some organisms, cell division is a way for reproduction, okay? So that's the last part is not true for humans because humans reproduce sexually. You need two individuals, each contributing half of the genetic materials. But for some organisms, they reproduce asexually, okay? Only one individual is needed, and they do so through cell division. Okay? So the cell division is called mitosis, which is a form of asexual reproduction. So what is asexual reproduction? Basically, no sperm and eggs are involved, right? There's no fertilization happening. And the advantage of that, it's you only need one individual. Sometimes it's hard to find a partner, right? In the wild, like think of like a whale in the ocean or something, right? Like how do you find another whale? It's such a big, vast ocean, right? So, it's a challenge, but for some organism, they don't need to find a partner. It's just one individual needed. It's fast and it requires less energy. Okay. A bacteria is able to divide every 20 minutes under ideal condition compared to like humans. If we carry a baby, it takes about 10 months, right? Okay. So and a lot of energy, of course. So that's the major advantage. The major disadvantage is low genetic diversity. Okay, if you are just making copies of yourself, then all the offspring will be genetically identical to you. Okay, which could be a problem because if there is some kind of genetic defect, that defect will be in every single offspring, right? Okay, so that could be a problem. But later, as we will see, being able to divide rapidly is actually a way to introduce genetic diversity in the population. Okay, so even though all the offspring you produce are genetically identical in most cases, but if you're doing it fast enough, because of mutation, which is something we'll learn, it's a great way to introduce genetic diversity. So some organisms that can do asexual reproduction, sea star or starfish, some people call it. Did you know if you cut a starfish in half, both will grow into a starfish? Okay, so that's asexual reproduction. Okay, you buy one, now you get two, right? It's not the same as an earthworm, right? Earthworm, you cut it in half, only the head will grow a tail, right? The tail is not going to grow the head back, right? Okay, so that's regeneration. That's not reproduction. Amoeba, we talked about amoeba before, it splits, right? A lot of plants, they reproduce asexually. If you grow any kind of plants at home, you can break a piece off and give it to your friend, right? Okay, like aloes. It's like I have so many aloe plants growing, like I try to sell them online. Nobody buys them anymore. And then we have this thing called hydra. It's a microscopic thing and it grows by budding. So like a little baby just start growing off itself and then it snaps off. Okay, and this is another form of asexual reproduction. Okay, you don't have to memorize any of these things. Okay, good. But you should know what asexual reproduction is. Okay, just want to show you this picture again to help you remember. DNA is double helix, but we do not have naked DNA in our cells, right? We have to package them first before we can fit it into the nucleus. So when a cell is not dividing, the DNA exists in a form of chromatin, which is just DNA wrapped around proteins. Okay, we just call them proteins. It's called histone proteins, but we'll just say proteins. Now, when you need to divide the cell, then you need to pack up the chromosome, the DNA even further, and that's when you get chromosome. So chromosome and chromatin are basically the same stuff, but one is way more compact compared to the other. The most compact form, the most condensed form is the chromosome. Okay. So I asked you many times already how many chromosomes we have in the body, right? And by now, you should know it is 23 pairs, okay, which is 46 chromosomes, okay? So you get 23 chromosomes from the father and 23 chromosomes from the mother, and that creates 46 chromosomes. Of the 23 pairs of chromosomes, 22 pairs are called autosomal chromosomes, okay, or autosomes for short. These autosomes, these 22 pairs, codes for all the features of you, okay? Your hair color, your blood type, right? The kind of earlobes you have. Some people have like, detached earlobes. Some people have attached earlobes, right? You know what I'm talking about? Like if this is the face, this is like attached earlobes, and then like this is like detached earlobes, right? So that's genetically determined. Some people have 5 fingers. Most people have 5 fingers. Do you know some people have 6 fingers? Okay. Some people are born with 6 fingers or sometimes 6 toes. That's genetically determined. So all these things are part of the autosomal chromosomes. Okay. The 23rd pair is the sex chromosome. If a person is XX, then that's going to be a female. If it's XY, then that's going to be a male. Okay. So I want to ask you, who determines the gender of the baby? The father or the mother? Hmm? The father, right? Okay, because the father is the one that has the Y. And when the father produced the sperm, half the sperm will get the X, the other half will get the Y. Depending which one fertilizes the egg, then you will have a daughter if it's XX, or a son if it's XY, right? Okay. My grandmother is very traditional. So in her mind, she thinks is the woman that is deciding whether it's a boy or girl, right? So I have 2 daughters, I told you already. So every now and then my grandma, she's like 90 something, right? She would sit beside me and then whisper in my ear, why is your wife not giving you any son, right? And then I will have to tell you, I will have to tell her, it doesn't work like that. It's actually my fault. And she's like, no, 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 no. I know. I have many sons. I was like, okay, okay. We'll just leave it at that, right? So it's the male that determines whether it's a boy or girl. And when I say determines, I don't mean like they could like deliberately think which one to choose. It doesn't work like that. It's just by chance. Okay, of course. And whether you have, let's say you have this is another thing she said for real. Okay, so you have 2 girls now. The next one will be a boy. And I was like, no, it also does not work like that. If you flip a coin and you get heads for 4 times, what's the chance you're gonna get a tail on the next one? 50%, right? Okay. Each event is independent of the previous events, right? Okay. Anyway, just fun stuff. So we have 46 chromosomes, 22 pairs. But it turns out the number of chromosomes an organism has is not related to its size or anything like that. Okay. Goldfish, for example, has 100 chromosomes. And some of the plants, they have up to thousands of chromosomes in some cases. And there is seemingly no correlation between the size of the organism and how much genetic material it has. Okay. All right, so we're gonna talk about the cell cycle. Okay? The cell cycle is mainly separated into 2 key events. Right? We have what's called the interphase. So this is roughly about 90% of the time for most cells. Okay, they spend their time in interphase. And there's nothing like really special going on during the interphase. They are just performing its normal function. They are, if it's a muscle cell, it's going to be contracting. If it's a liver cell, maybe it's detoxifying. It's basically performing its normal function. Okay, so 90% of the time it's going to be doing this. And it's growing. And eventually, when it gets too big, then it will switch to the cell division, the mitosis. Okay, so this is roughly just 10%. Now, obviously, these are very generic numbers. They are just average. Some cells divide much faster than other cells. Can you think of a type of cells in your body that divides like really rapidly? Like you almost get brand new sets of these cells every 3-4 weeks. Yeah, skin cells, right? Okay, skins, you get new skins replaced very, very rapidly. Okay, some things they divide very slowly or don't divide at all. Okay, the heart, the cells don't divide in the heart. That's why there's no such thing as heart cancer. If a cell don't divide, then it's not possible to have uncontrolled cell division. Right? No such thing as heart cancer, right? And then some cells have more regenerative capability than others. Did you know if you donate half, one third of your liver, right? You will grow it back. Okay, so you can donate to someone who needs the liver, and then yours will come back after a while. So very remarkable regenerative capability there. Whereas if you lose a finger, finger's not going to grow back, right? So this is the cell cycle broken down in more details. I am actually going to copy this over. You know what? Let's just draw it from scratch, and then it will be the last thing we do. Okay, and I'll send you home. Cell cycle. Okay, so this like whole thing is interface. 90% of the time on average, doing its own thing, growing, gathering resources for the ultimate division. So there are a few key events that will be important in interface. Okay. So over here, we have what's called the G1 checkpoint. And then over here, we have the G2 checkpoint. Okay, it's like in Canada, if you want to get a driver's license, you need to pass the G1 and the G2. So before you can do the cell division, the cell has to pass G1 checkpoint and G2 checkpoint. That's actually what they're called. And sandwich between G1 and G2 is the S phase. Okay. S stands for synthesis. But what's more important is that the S phase is where DNA replication takes place. Okay. Remember, before you divide, you have to copy all your genetic material. So all that stuff we learn about helicase, gyrase, so on and so forth, that happens in the S phase. So that precedes the actual division. Okay. So before you can copy your DNA, you need to check a few things. That's what G1 checkpoint is. Okay, so there are a couple of things that we can check. Number one, the quality of DNA. For example, any damage, any mistakes, right? Like it's an A pairing with a C instead of A pairing with a T. Right? If there are mistakes like that, you got to fix it. Okay. If there are damages, you need to fix it as well. We need to check the length of telomeres. Are they long enough? If it's critically short, it should not be allowed to go through another round of DNA replication because you're going to cut it even shorter, right? So you must check to see if the lengths are okay before you give them the go-ahead. Now, you need to check the cell size. Is it big enough? If the cell is really small and you divide it, then you end up with two tiny cells. They might not be able to function properly. So it has to be big enough. All right, cell size big enough. All right. And you also want to know if the cell has enough resources. What kind of resources? For example, nucleotides. We're going to be replicating the DNA. We're going to need a lot of ACTGs, billions of them. Do you have enough? So where do you get your nucleotides in the body? How do you get nucleotides into your body? From eating, right? When you eat the food, the protein gets broken down into amino acid, right? And then the DNA that's in the food, your food is like come from living things, right? Okay, so that's going to have DNA in it, and your body breaks it down into the nucleotides, which is going to be absorbed by the cell, right? So do you have enough resources? Okay, so if everything is okay, no DNA damage, or if there is damage, you try to repair it, okay? And if you're able to repair it and the lengths are good, the size are good, then you will allow to go to the DNA replication step, okay? If not, if the cell fails G1, then it will trigger apoptosis or cellular, or which is a growth arrest. Okay? Senescent. Senescence. Remember apoptosis? What's apoptosis? Cell death, that's right. It's caused the lysosome to release all the enzymes. We better kill this cell, okay? Because there are critical errors that cannot be repaired, okay? If the telomeres length are just like too short, then you don't have to kill the cell, okay? The cell can continue to do its work. So it's just going to enter growth arrest. Growth arrest means that you can't grow anymore. You can't divide, okay? You can keep doing what you're doing, okay? But we don't have to like destroy you. Okay? It becomes senescent, okay? And if the cell size is not big enough, then no big deal. You just wait longer. Maybe the cell will get bigger, right? So like the point is, if you fail G1, you can reproduce yet, okay, or ever. And then if it pass, then you're going to do DNA replication. After DNA replication, okay, so for example, you have chromosome 1 from dad, and then chromosome 2 from dad, chromosome 1 from mom, chromosome 2 from dad. Of course, 3, 3, 4, 4, 5, 5, all the way to chromosome 23, 23, right? Okay, so DNA replication is gonna basically double all of it. So now you have 2 chromosome 1 from dad and 2 chromosome 1 from mom, and so on and so forth. Okay, so each of the genetic material is going to get double. Okay. We are almost there. Stay with me. So afterwards, after you copy the DNA, we're going to have another checkpoint, right? What are we checking for this time? Any ideas? There's just one key thing we need to check for. It's like after you do a test, you have to check your test, right? What are you checking for? Mistakes, that's right. That's what this is. Okay. Check for mistakes and also damage. Okay. If fails, G1 or G2, I guess. Then again, even we copy the DNA, but looks like, there are some major issues here, right? We're not going to be able to move forward. We have to kill the cell. And if everything's good, then you prepare a little bit more. And then finally, you're going to do the mitosis, the actual cell division. Okay. So this is where we are moving next time. Next Monday, we will do the actual mitosis. There are 4 steps in it. And then we will look at what happens if the cell cycle checkpoints fail. Okay? Look, the checkpoint is supposed to stop the cell from moving forward if it has mistakes and damages. But if the checkpoint fails and the cell is allowed to keep on dividing despite having mistakes, then we have a problem because those cells will now continue to grow and accumulate even more mistakes, and that eventually could lead to cancer. Okay, so don't forget to change your clock this Sunday. You have an extra hour. If you're new to Canada, it's a thing that we do every fall and spring. And print lecture 7 for Monday. Okay, thank you guys.